So we're gonna continue with this. This is talking about how does the body control urine concentration and volume, and we pointed out that our kidneys work really hard to keep the osmolarity of the fluids in our body at 300 milliosmoles. And it does that through controlling not only volume of urine, but also the concentration of our urine, because that's going to affect volume and concentration of other bodily fluids. You've definitely seen that. How does it do that? How does it control urine concentration and volume? Well, it does it through this amazing situation called a countercurrent multiplier. And this basically uses juxtamedullary nephrons to create a osmotic gradient to help concentrate urine that's leaving the body. So if you think back about when we talked about the two types of nephrons, we had cortical nephrons, which were the little short nephrons, and then we had the juxtamedullary nephrons that basically had these really long loops of Henle, and they dipped way down into the renal medulla. So if you remember back then, I said, well, these accounted for only about 15% of the nephrons as a whole, and they were responsible for creating concentrated urine. So how does that happen? This is going to happen through this countercurrent multiplier and the creation of a medullary osmotic gradient. So what do we mean by that? So basically, if you look at the kidney, we take a kidney and let's see, here's my renal pelvis and here's my, ureter here. So we know kind of the outer part of the kidney is going to be the cortex, and then deeper towards the inside is going to be the medulla. So basically what we see is the cortex of the kidney is about the same osmolarity as fluid in the rest of the body. So it's going to be sitting at about 300 milliosmoles, okay? This medullary osmotic gradient means that the osmolarity is going to increase as we travel deeper into the kidney. It's going to keep increasing in terms of concentration of solutes up to the point where we get to like 1200 milliosmoles. And the same thing would happen here. So we'd start off at the cortex at about 300. The deeper we go, the increased concentration or osmolarity we see, and we ultimately get up to about 1200 milliosmoles. So our osmolarity is increasing as we move from cortex to medulla, right? Okay, so if you recall, those juxtamedullary nephrons were the nephrons that had those really long loops of Henle. So we would have, kind of our renal corpuscle here, and then a really long loop of Henle that's going to dip way down into the medulla. And so that's kind of what we see here is this nephron here with this really long loop of Henle. It's going to dip way down into the medulla and then come back the other side. So what's going on in this process? As filtrate enters the loop of Henle, it is at an osmolarity of 300 milliosmoles. It's the same as all the other fluids in the body, okay? So filtrate's going to move down. It's going to start traveling down the loop of Henle. And what happens is we know that aquaporins are present in the descending limb of the loop of Henle. So water is going to diffuse out of the descending limb of the loop of Henle. And as water leaves, it's going to further concentrate the solutes that are left inside the tubule. So as water's leaving the whole time that tubule is traveling down, water's leaving, and it's concentrating the solutes that are left inside the tubule. So we see our osmolarity increase as we travel down. So it goes from 400 to 600 to 900, all the way down to 1200 at the loop. And that's what I've shown you here. This is 1200. So this is kind of where our loop is. This is our like super concentrated part of the renal medulla. Okay, so all of our water's left, and we're left with a super concentrated filtrate inside the tubule. Now, what's happening in the ascending limb, it's not permeable to water because there's no aquaporins present, but we can pump salts out of the tubule. So at this point, we've got increased concentration of solutes inside the tubule as compared to outside. So we're going to actively pump salts from the tubule into the extracellular fluid. So as a result, we're going to have increased concentration of sodium chloride outside the tubule in that extracellular fluid. Okay? It doesn't just stay right here, it's actually everywhere. So even on this side, we're going to have increased concentration of sodium chloride. Okay? And so as a result, we're going to have decreased concentration of water, increased concentration of water inside the tubule. So the next kind of batch of filtrate that makes its way through is going to pump out even more water. It's going to further concentrate that filtrate that's left inside the tubule. And as a result, on the ascending side, because it's permeable to sodium chloride, it's going to pump out our solutes, okay, sodium and chloride and potassium. Okay, so we had left off previously with talking about how this countercurrent multiplier that is created by the juxtamedullary nephrons is able to allow nephrons to produce varying concentrations and varying volumes of urine. So I kind of left off here. So our ascending limb is responsible for pumping out solutes, and that's what we see there. And it actually pumps it out to the point where if you look at the osmolarity of the filtrate that's left inside the ascending limb, and as we head into the distal convoluted tubule, it's very low. It's actually much less concentrated than just your normal bodily fluids, which would typically be around 300. So here we're down to about 100 milliosmoles. And so in that case, in this scenario, what would happen is if we are overhydrated and we've got plenty of water, then our body is not going to release antidiuretic hormone. So we're going to say no ADH. And so we know that hormones have their effect on the distal convoluted tubule and the collecting ducts. So when we have no ADH released, when we are fully hydrated, and in fact even overhydrated, we're not going to have any ADH released. So there's going to be no aquaporins present in the distal convoluted tubule and collecting duct. So that urine is going to make its way down the collecting duct, and what we're going to see is that our urine is going to be very dilute. It's not concentrated at all, so this is going to be a very dilute urine. And then it's going to be a lot of it because we're not pulling water out of the tubule. It's going to be a high volume of very dilute urine. And so that's what we see when we could even add up here overhydrated, and we have no ADH that's being secreted. Okay, so that's how our nephrons will create dilute urine and lots of it. So let's change the scenario a little bit, and let's show how. Let's get rid of. Let's get rid of this and we'll start this over again. So if we had the scenario where we were out in a desert, or let's say you're running a 50K and it's the middle of summer, and you're dehydrated. Okay, so when you are dehydrated, your water volume in your body is going to decrease. And that means that the solute concentration is going to increase. So your body is going to react to that, and you've got chemoreceptors that are going to measure the osmolarity of your bodily fluids. And it's going to say, oh, you're dehydrated, so we need to release ADH. So in this case, let's say that we are dehydrated. As you can tell, the mouse writing on the screen there's a learning curve. So we're dehydrated. And so what happens with that is that we're going to have increased ADH secretion. And we know that ADH has its effect, or exerts its effect, on the distal convoluted tubule and collecting ducts. And it does that by dropping in aquaporins. And so that's what I'm going to draw here. Let's say that these are aquaporins. Okay, so when we have ADH released, we're going to have aquaporins present in the distal convoluted tubule and collecting duct. So the urine is still leaving this area at pretty low concentration. It's still about 100, okay? But what's going to happen is as it travels through the collecting duct, we are going to be pulling water out of the tubule. The whole way down, we're actually going to be reabsorbing water. So the more water that we reabsorb, the more concentrated the urine that's left inside the tubule is going to become. Okay, so as we're pulling out water the whole way down, this osmolarity inside here is going to increase, and then we're going to pull out even more water, and it's going to increase, and we're going to pull out even more water, and that urine is going to increase even more in its concentration. So as a result, when we're dehydrated and we have secretion of ADH, we are able to concentrate our urine, so it's going to be highly concentrated, and it's going to be not much of it, because we've reabsorbed a lot of the volume of the urine by reabsorbing all of this water. Okay, so it's going to be a concentrated, decreased volume of urine. Okay, and so that's how this countercurrent multiplier establishes the medullary osmotic gradient so that we are able to increase water reabsorption in the presence of aquaporins in that distal convoluted tubule and collecting duct. Okay, it creates that gradient, and the nephrons are going to use that gradient in order to concentrate the urine. Okay, this is just the last thing on that. The vasa recta, we know that that's the specific peritubular capillary beds that surround the juxtamedullary nephrons, and that's really where all of this magic is happening. And the vasa recta basically just preserves that gradient. It doesn't undo it. So the kidneys work really hard to create that gradient, and the vasa recta, those capillaries, are so permeable to the concentrations of water and solute around them that they basically are the same. So if you look at the concentration of solutes within the vasa recta, it's fairly similar to the concentration of that extracellular fluid around it. So we have this kind of, free exchange. And so that's basically what we mean. You might see that written like in your textbook, but the fact that the vasa recta preserves the gradient, and that's what that means. So they're highly permeable, and they basically stay, they're the same osmolarity in the capillary as the extracellular fluid. Okay, so what do we have in urine? So it's mostly water, like most other things in the body. It's like 95% water, and about 5% solutes. So for instance, uric acid, creatinine, some ions, so sodium, potassium, calcium. Um, there really shouldn't be a whole lot in your urine. This kind of pairs well with the lab that we did. Sometimes having a high concentration of solutes or protein in your urine may indicate pathology, or it may just be a result of something completely normal, like a really hard workout. So it really just depends. And when you have patients come in, it's important to take a history and figure out what they've got going on. The color of urine would typically be clear to pale yellow. It would have a slight odor that would be normal. It's going to become more odorous the longer it's exposed to air. And urine is slightly acidic and typically has a pH of about 6. So again, diet changes can affect pH. If you eat a diet that's super high in protein, it's going to become more acidic. If you're more vegan, then it's likely that you have urine that's more alkaline. In terms of other urinary anatomy, we talked before about ureters, and so these are the tubes that are going to drain urine from the renal pelvis down to the urinary bladder, and they do have that layer of smooth muscle, and so we see peristalsis happen in the ureters like we see in other kind of tubular organs that we've talked about this semester. So it's not just gravity pulling urine down, it actually is propelled through peristalsis on its way down to the urinary bladder, which is basically going to be a muscular sac for urine. It's going to hold it until you say that it can come out of your bladder. And what we see in the urinary bladder is folds in the wall of the bladder, and that's called rugae, just like the stomach. So it's able to distend and, excuse me, fill with urine before we let it out. The walls of the bladder do contain a layer of muscle, and that's called the detrusor muscle. And that's what's going to contract when you urinate in order to push that urine out. So the urethra is the muscular tube, and we can see that here. So this is going to be the urethra on the male anatomy. This is the urethra on the female anatomy. And so that's just the conduit to take urine from the urinary bladder to the outside of the body. And this really kind of highlights why females are more likely to get bladder infections, because we've got these shorter urethras, and so bacteria from the outside of the body can make its way a little more easily into the bladder. Males have such a long urethra and are less likely to get bladder infections. And another difference too, since we've been over reproductive anatomy, is that the urethra in males is a shared space between reproductive and urinary system. In female, it's a dedicated urinary system structure. And then the last thing is the trigone, and I know we've talked about that before, but that's basically that triangular shaped region between the insertions of the ureters from the kidney and then this internal urethral orifice, which is basically where the urethra is emptying the bladder. It forms this triangular shaped region inside there. Urinary system has two sphincters, so we see two sphincters associated with the bladder. First is the internal urethral sphincter, which is right where the urethra empties from the bladder, and that's smooth muscle. It's under control of the autonomic nervous system, and it's involuntary. And then the external urethral sphincter is skeletal muscle. This is external. Here's your internal, and is voluntarily controlled. So this sounds very familiar. I mean, this is similar to the setup that we had for the anal sphincters, internal and external anal sphincters. So basically the same thing there. And the way that comes into play is when we talk about the process of micturition, which is the act of emptying the urinary bladder once it becomes full. And so what we see is urine is constantly, being conveyed from kidneys down to the bladder, and the bladder is going to stretch, and the wall of the bladder, the interior of it, is going to have stretch receptors that's going to detect that stretch and send afferent or sensory impulses up to the brain. It's initially relayed to the thalamus, which we know is the relay station for sensory information from our body. And thalamus is going to relay it to our sensory cortex so that we have a conscious awareness of a full bladder, as well as to visceral sensory areas of the brain. So we do have voluntary control over this external urethral sphincter because it's skeletal muscle. So we can, decide when it's a convenient time to go to the bathroom. And we're getting signals from autonomic nervous system, namely parasympathetic division, saying, okay, here's this information that we're getting. Your bladder is full. You need to use the bathroom. And then we have conscious kind of override of that information. So when we decide that it's a good time to use the bathroom, the detrusor muscle inside the wall of the bladder is going to contract. We are going to consciously relax this external urethral sphincter, and as a result, that's going to allow the internal urethral sphincter to open, and urine is going to leave the body. That's kind of the overview. I also included this kind of pathway too, but this basically walks through the same thing. So the three things that have to happen as part of micturition is you have to have contraction of the detrusor muscle. So we get stimulation of detrusor muscle. We get voluntary relaxation of the external urethral sphincter, and we get relaxation of the internal urethral sphincter as a result.